think we're going to get started. Welcome to the genetics session. I just want to make one announcement before we start. Immediately after this session at noon, the results from the student paper and poster judging are going to be announced in the Regency, which is down the hall. So noon right after this session, head down there. Our first talk of the genetics session will be Nicolas R, and she will be talking about adaptive divergence in Atlantic cod.
Okay. Do I need to use the mic? Or?
You don't have to if you don't want to.
Can you guys hear me okay? Okay, I'll skip it then. Okay, great. So I'm going to be talking about some recent work we've been doing using whole genome sequencing to look at the population structure in the remnant spawning groups of the very depleted Atlantic cod in US waters. And I think this work both illustrates some interesting new insights about local adaptation and diversity in cod, but also more broadly, it illustrates how the incredible advances in DNA sequencing technology that make it possible to now screen whole genomes, even for large population samples, how that's really providing a whole new level of resolution in population genomic studies, and I think it's likely to provide give us surprising findings in many different species. So this cod project is a collaboration between my lab and Adrienne Kovach at the University of New Hampshire, and the work I'll be showing today was led by her postdoc Gemma Glucas in collaboration with my grad student Nicolas R. So we all know that cod for centuries formed the basis for an enormous fishery in New England, but back in the early 90s there was a massive collapse in this fishery. And as you can see here from the landing statistics and estimates of biomass are showing the same pattern of this dwindling trend ever since. So this lack of recovery, even now, more than 25 years later, making people question whether we need to consider how we are managing these species. And what we are particularly interested in is whether the current definition of stocks that's being used for management is really appropriate for how biological diversity is found within the cod populations that remain. So by now, it's fairly broadly recognized that biodiversity tends to promote resilience in ecosystems. So for example, in coral reefs, species diversity have been shown to promote ecosystem health. But also within species, there can be really important diversity, sometimes called biocomplexity. So that can be, for example, structuring of populations into local self-sustaining populations, subpopulations that may be adapted to local conditions to show different ecotype variation. And this phenomenon has been most famously studied in Alaskan sockeye salmon, where it's been clearly shown how within population diversity can really buffer systems against both environmental perturbation and also fishing pressure to cause more sustainable fisheries long term. And in a simple sense, we can also just think of if we have a big open ocean area that happens to have three more or less isolated populations, if we manage that as a single unit and we just set one quota for this whole area, it might be that fishermen will tend to exploit just one area that's most convenient to them or where it's easiest to catch. And you can see here, of course, that in this scenario, the simple scenario here, we'd see really large overexploitation from this population that's in the most popular, maybe near-shore area, and underexploitation in populations that are perhaps further offshore. So if we can get a better match between the units that we generate management actions for and the actual biological units in the system, we can expect much more sustainable fisheries overall. So how does that apply to cod? Cod is distributed on continental shelves throughout the North Atlantic, and lots of prior work has shown that this species shows very extensive genetic differentiation across this range, and also quite extensive temperature-associated adaptation and large divergence in different ecotypes. So for example, inshore and offshore groups of fish. And if we zoom in on the US waters here, so for many years, cod in US waters have been managed in just 2 units. So there's been a Gulf of Maine stock and a Georgia's Bank stock. That includes the southern New England waters. But recent interdisciplinary work that has included some early genetic studies, outlet microchemistry, tagging data, phenotypic data, and a whole lot of other evidence have suggested that this two-stock model might be inadequate, and that there's some evidence that within the Gulf of Maine there are actually multiple independent components, and that also that the spawning groups found down here in southern New England actually shows more connectivity with one of the components within the Gulf of Maine than with Georges Bank that they're currently lumped together with for management. But there's still a lot of uncertainty about this. So what we wanted to do with this project was to use genomic analysis to look in much greater detail at what the population structure is of the remaining actively spawning cod within all of US waters. And specifically, we also wanted to look at what are the particular genetic differences that we see between the groups. And this is where I want to make the point that genetics is not just genetics. So even if some study using maybe 10 or 20 markers, as we were able to do, I mean, that was all we could do just a decade ago. Even if those kind of studies have shown no detectable difference between groups, it doesn't mean that there is none, because genomes are really large. So typical fish genomes are range between something like 500 million to more than 1 billion base pairs. And different evolutionary forces can have different effects on genetic variation across various regions of the genome. So we can see contrasting patterns. And you might have seen this graph before. So this is showing the drop in DNA sequencing prices since the beginning of the century. And notice that this is the logarithmic scale here. So basically this means that since year 2000 there's been a more than 300,000-fold reduction in DNA sequencing costs, so the cost of sequencing a base pair of DNA. So that opens up entirely new opportunities that mean that we can both screen much more of the genome, and we can do that in many more samples for a reasonable cost. So we've developed a very efficient technique to take advantage of these really cheap prices and do low coverage whole genome sequencing of large population samples. So basically what we do is that we extract DNA from individual fish and attach a little barcode so we can recognize the DNA from each individual. And then we pool it all together from maybe hundreds of fish and sequence about the genome about 1 time over for each fish. So that we can do for about $50 per sample. So that's making this technology a real possibility even for research projects with pretty modest budgets. But the downside is, or the limitation is, that with current DNA sequencing techniques, error rates are so high that just sequencing the genome 1 time over, we can't reliably call individual genotypes at particular variable sites. But we can take, when we have large population samples, and we can take the uncertainty into account statistically and then integrate over that to get reliable population allele frequency estimates. And in the case of the cod, we were able to do this for about 11 million SNPs or variable sites in the cod genome. So you can see that's really many, many orders of magnitude increase in the number of markers than what people used to use. I mean, what was the only possibility only a very short time ago. So the samples we looked at in this project were from, we tried to get actively spawning fish from most known active spawning grounds in the Gulf of Maine or in all of US waters. And then we also included a couple of Canadian samples for reference. And especially interesting is this area here within the Gulf of Maine, where in very close proximity there are different groups that spawn in the winter and in the spring. And we were able to get samples from both of those to include. So in total we looked at more than 300 individuals from 20 different locations. And the first results I'll show you are comparisons between this furthest away Canadian samples and some representative locations within US waters. So what we're looking at here are each of these horizontal panels here are one of those populations that we're comparing the Canadian sample to. The horizontal blocks are linkage groups or chromosomes in the genome. So basically the data are organized along the stretch of the genome on the x-axis. And on the y-axis we have FST, or a measure for the genetic differentiation. So basically how different are allele frequencies between the Canadian sample and the particular population that we're comparing it to. And each dot here is the average across 15,000 base pairs in the genome. So if we just first zoom in on the bottom 4 here, the first thing that jumps to mind is that the majority of the dots are right here down along the 0 line. So that's basically suggesting that the vast majority of the genome is showing really minimal differentiation between the Canadian and the US populations. But then what jumps out are, of course, these blocks here that are showing very highly elevated levels of differentiation. So this is actually quite, the numbers here are actually quite, this is like really, really noticeable genetic differences. But you can see that if we in a study, we're just using, for example, microsatellite or some other marker that's randomly distributed across the genome, even 20 sites or something, we could very easily completely miss these regions that are showing the high differentiation and just get an impression of almost no differentiation in this system here. But these blocks here are a little bit odd, and what is causing that is a well-described, a well-known phenomenon in cod, which are chromosomal inversions. So it's basically very big chunks of chromosomes that have at some point been swapped around, and then both directions are still found. So that's a polymorphism that's found in the population. So these inversions here then link together hundreds of genes in one block where there's no or very little recombination, so they get inherited as a block. And those kind of inversions can be, because they have so many genes, they can have lots of functional effects. So therefore they're likely to be involved in local adaptation, as seems to be the case here, because that's where we're seeing the big differences between the populations. But another thing to note is, in addition to these big blocks, we also see a couple of really narrow peaks, like this one here. So that shows up even more clearly when we compare all the populations to one of the spring spawners, because it turns out to be a peak that's causing consistent differentiation between all the populations that are winter spawners and then the spring spawner. And that's really interesting because the differences here, these are averaged, but they're still getting pretty close to 1 within a big window. So that means that in almost all the spring individuals we find only one variant, where in all the winter individuals we find a different variant. So that's a really striking consistent genetic difference against this baseline of basically no differentiation in other parts of the genome. And what's particularly interesting is that these peaks that are quite narrow in the genome, around 50,000 base pairs, are located right over several genes that are known to be involved in gamete maturation, so follicle stimulating hormone and estrogen receptor. So this makes a lot of sense that this is a really important region that has to do with adaptation to different spawning seasons in cod. But what is it that can cause these kind of different levels of variation in the genome? So generally, genome-wide effects, so genetic drift and gene flow, will have similar effects throughout the entire genome. So when we're seeing throughout most of the genome almost no differentiation, that can either mean that there's a lot of connectivity between the populations, or it could also be the case that there actually isn't much connectivity right now, but the population sizes are just so big that there hasn't been time for them to diverge. But in contrast, selection we expect to have these really localized effects because that's targeting specific genes, specific functional variants. So that's what's driving these localized peaks in differentiation. So even if genome-wide, whether there's connectivity or not, the fact that we're seeing these consistent, like in multiple independent comparisons, we're seeing this very high differentiation at what appears to be functionally important regions is suggesting that this is some functionally important diversity that we want to protect. And it's kind of interesting because the different parts of the genome tend to group populations differently, so the different regions under selection. So in this heat map here, similar, so here we have different genomic regions listed here, and then we have the different populations listed in the columns here. And there are a couple of regions that are, and the color of each cell is indicating how similar two populations are. So basically more similar colors means more similar genetic signature. So there's one group of outlier regions that tend to just group the two Canadian samples from the rest. Then there's another group that, another set of genomic regions that group together the Canadian and the spring spawning samples that we have. And those are regions that in other studies have been associated with local temperature, salinity, depth, and also ecotype behavior. Then we have those particular narrow peaks that separate out our spring and winter spawners. And then we have a small region that's really separating out the two most southern locations that we have that's been associated before with thermal tolerance, and there's some heat shock proteins in there. So overall, it's a pretty complex and multifaceted signature of different selective pressures acting on different regions of the genome in different populations. But overall, suggesting that there is definitely quite a bit of biocomplexity to conserve in the system. And if we look at the overall pattern, so this is a multivariate analysis across all our 11 million SNPs. The general pattern that we're seeing is that we're getting the Canadian samples really separating out, and we're also getting this inshore spring spawning component separating and Georgia Spain. But then what's interesting about what's unexpected is that the two southern New England samples are really grouping out separately from the rest of the winter spawning samples that we have out here in the Gulf of Maine and out towards the Georgia Spain. And then another interesting thing is that we included samples. So up here in the eastern Gulf of Maine, there used to be very active spawning, but there isn't anymore. And we included some samples of non-spawning fish from up there to see whether it might be remnants of some population or if it's now just been starting to be occupied by other populations. And it looks here there's some tendency for them to separate out in their own piece, which could suggest that it's a unique population. But we're looking into that much more closely in follow-up work. But basically the conclusion is that compared to this two-stock model, our work here is really suggesting that there are at least 4 different components that should be considered in the management, and that some of them are even kind of spanning across the current stock boundaries. So Adrienne Kovach and I are currently members of a working group that NOAA is organizing to revise the COD structure, where it's getting input from all different disciplines. So we're trying to make sense of this and make that inform management. So I guess I'll stop here.
Okay. We have a couple minutes for questions. 2 minutes.